Every living cell, from plants to humans, emits faint pulses of light. They are called biophotons, ultra-weak photons invisible to the naked eye, yet possibly fundamental to life itself. Biophotons are not ordinary light. They are ultra-weak, coherent emissions in the visible and ultraviolet spectrum. Detected only with the most sensitive instruments, they may act as messengers, regulating biological processes in ways modern science is only beginning to uncover. The story begins in 1923, when Russian scientist Alexander Gurevich performed a now-famous onion root experiment. He observed that when two onion roots were placed close together, one root seemed to stimulate cell division in the other, not through contact but through an invisible radiation. Gurevich named it mitogenetic radiation. He believed cells were communicating with each other using ultraviolet light. If true, it meant that biology had a hidden light language. His claim stirred both excitement and doubt. Some researchers replicated the effect, others failed, dismissing it as experimental error. Without sensitive tools, the phenomenon seemed impossible to prove. As the decades passed, Gurevich's discovery slipped into obscurity. The scientific world moved on, labeling it a curiosity without solid proof. But science caught up. In the 1950s and 60s, new instruments, photon detectors thousands of times more sensitive than the human eye, confirmed Gurevich's hunch. Plants, bacteria, animal cells, even human tissue were all found to emit faint photons, continuously, as if alive with light. By the early 1970s, the idea of biophotons had returned, no longer myth but measurable fact. The stage was set for a new pioneer to take Gurevich's vision even further. That pioneer was Fritz Albert Popp, whose bold experiments would spark a revolution in biophoton research. His work would suggest that DNA itself is a source of living light.